on Monday. I'm a little bit slow on grading the first worksheet. I'll get it done over the weekend. But I already took looked it over, and I want to warn you about your graphs, okay? Because some of you are not reading and understanding the graphing rules in exercise 1. So you always have to go back to exercise 1. I have a whole list of detailed graphing rules, okay? And so you need to follow those rules. Any graph has to occupy at least half a page, okay? So if you have a page and you have a little graph like that, that's not half a page, okay? Way too small. And so you need to expand it out so that it's big enough. Now for histograms, you really don't need grid lines, but for other types of graphs, yes, you need to have lots of grid lines, so that it's easy to read, okay? And so just make sure that you go back and look at those graphing guidelines. Usually in lab, I go over each and every one of those, but for the sake of saving a little bit of time, because they were tight, you have to finish at the end of the lab, I didn't go over them, okay? But if you have any questions on them, what I mean by those guidelines, you need to talk to me, okay? So I have office hours. I had office hours on Sunday through Zoom, and today I have office hours. So if you have any questions about how those guidelines are supposed to go, please look at them, all right? Okay, and some of you didn't finish exercise 3 yesterday, and so before you leave today, you really need to turn in that worksheet, okay? All right, so let's carry on. I believe this is where I stopped, is that right? Okay, that's good, because I'm teaching 2 classes, and I'm teaching majors biology and baby biology, and we're talking about the same thing, okay? And that class is right after this one, so I never know where I stop. Okay, so talking about the traffic, okay, so things are made in smooth and rough ER, shipped by vesicles to the cis side of the Golgi, where they are modified and sorted, and then from the trans side of the Golgi through transport vesicles, then they are transported to the proper place where they go. And that could be the plasma membrane, that could be a lysosome, that could be a vacuole, that could be back to the, even to another part of the Golgi, or back to the smooth ER, or the rough ER, okay? Some part of the endomembrane system, but not peroxisomes, not to mitochondria, not to chloroplasts, okay? Because those are not part of the endomembrane system. And when this movement takes place, those 2 faces of the membrane keep their original orientation, okay? And anything that's put into them, inserted into the membrane, has to be inserted in a fashion that they will be in the correct orientation when they arrive at where they are to function. And so that is topological equivalence, okay? All right, so these membranes are semi-permeable. So if we stripped out all the protein, and we're just looking at a phospholipid bilayer, that is what is semi-permeable, okay? Just due to its chemical nature, certain types of solutes can easily pass through, others with difficulty, and others not at all. And so what are the factors? When I put factors up here affecting the permeability, I'm talking about not factors of the phospholipid bilayer per se, but of the diffusing solute, okay? What is the nature of the solute that affects it? So these are semi-permeable, and so some of these factors is size. Because if anything is going through a phospholipid bilayer, it has to push phospholipids aside. And that takes force, takes energy. And so if it's a big bulky thing, that's too much too much to do. So something that's big can't get through. Something that's real small can push the phospholipids up just a little bit aside and be able to cross. Okay? But also the polarity of that solute makes a difference. So the more non-polar it is, the easier it is to cross. The more polar it is, the harder it is to cross. Okay? And charge. Charge. If it is, now this is the extreme of polarity, okay? Because anything that's polar has partial charge, okay? Partial negative on oxygen, partial positive on nitrogen, and so forth. Partial positive on hydrogen, and so forth. But this is the extreme. This is a whole full charge. Now, we're talking about the combination of all three of those with regard to the solute, okay? Not just only one or the other, but all three. And so things that can usually cross pretty easily are relatively small nonpolar molecules. They are lipid soluble. They are lipid soluble, and therefore they can, they're hydrophobic enough, and therefore they can dissolve in the hydrophobic interior of the membrane. All right? And so this is the membrane. So things like carbon dioxide, oxygen, molecular nitrogen, these are all relatively small and they're non-polar. And you say, wait a minute, what about oxygen? Okay, now why would O2 be non-polar? Isn't oxygen electronegative?
It's because it's bonded to itself, or to...
Yeah, so we have two of them pulling electrons, and they're both pulling the same way. And therefore, there's no polarity there. To have an electrical dipole, you have to have one atom that is very electronegative, more electronegative than the other atom. Okay, but here, they're the same. Okay, so they're nonpolar. Okay, and so these things can pretty easily cross through the membrane. All right, not too much problem there. Relatively small, polar, uncharged molecules can. Okay, if they're small enough. Okay, so even now, if we put these two together, a small polar but uncharged molecule, if it's small enough, these can be even bigger than this. Okay, because they are completely nonpolar. Okay, so what I'm emphasizing here is the nature of the solute that can go through the membrane. We're not talking about these three characteristics independently of each other. It's a combination of them, okay? Now, usually water can't, okay? Because that's pretty polar. However, that really depends on the composition of the membrane. In some cases, if the lipid composition is such and such, water molecules can slip between phospholipids and cross. However, there's other pathways for water to cross, and I'll talk about that very shortly. In kidneys, there are some tubes and tissues that are completely impermeable to water. Okay, so those can be, it depends on, as I said, what the nature of the membrane happens to be. Okay, so these can cross. Okay, but not as easily. Okay, not as easily. Not as easily as something that's nonpolar. All right, so the diffusion across here would be slower. All right. Okay, so what cannot cross? However, large polar molecules. Okay, large ones like glucose and sucrose. They have too many polar bonds there. However, a lot of times membrane is still leaky. So there's a little bit of stuff that can get across, but we're talking about not much. And if we were to do something else with the glucose, okay, if we were to phosphorylate it, okay, put a phosphate group on it, okay, with the phosphate, there's two negative charges, plus 4 oxygens and the phosphorus. That will make the glucose not able to cross the membrane period, okay? And if we phosphorylate sucrose, it would not cross the phospholipid bilayer. So, and these are small, okay? These small ions, and that should be favorable, but having a positive charge is really rejected by the phospholipid bilayer. Okay? So ions cannot usually cross, all right? Unless there's a passageway to get across. Okay, so it's very obvious that cells have to have a way to move even large polar molecules and ions across their membranes. And so what are the transport mechanisms that are getting across the membranes? All right, one of these, simple diffusion, it requires a concentration gradient. Okay, in other words, the solute that we're talking about that has to diffuse has to be in disequilibrium. Okay, higher concentration on one side, lower concentration on the other. Okay, and so this is called passive. I'm going to correct some things because a lot of textbooks and websites will say that passive movement requires no energy. That is false. When they have anything that's in disequilibrium, that disequilibrium, unequal concentration, that is an energy source. And so here, the energy source for moving a molecule from this part across the membrane to that part is the concentration gradient. It is passive in that the cell does not have to expend its own energy to do so. Okay? That's what makes it passive. The cell does not use its own energy sources for the movement. All right? Directly. Okay, so what can pass through here, simple diffusion, across the phospholipid bilayer are small polar and lipid solutes. If they're small enough, they can diffuse across. If they're lipid soluble, very easy to get across until they get too big. Okay? If they're really big and lipid soluble, there's still some difficulty in crossing. All right? So I'll give you an example. Okay? Oxygen, nonpolar, generally across membranes, the plasma membranes of the cell, oxygen content outside is high, oxygen content inside is low. So there's a gradient, and therefore our oxygen can freely diffuse into the cell. All right? Now, for how long will this happen?
Until the concentration
Until we get equilibrium, right? Okay, until there's equal content on, equal concentrations on both sides. All right, but we have cellular respiration happening in mitochondria, and the reason we breathe for oxygen is for cellular respiration. So that oxygen is taken up by mitochondria, and it is involved in a bunch of chemical reactions, and therefore sort of consumed. No, they don't disappear. The oxygen just becomes something else, okay? And therefore, as long as the mitochondria are going along, this concentration gradient remains, okay? Now, the cell is not expending energy to bring in the oxygen. Even though there are energetic reactions happening here in cellular respiration, they are for some other function, okay? Not for transporting the oxygen in. And we're putting out carbon dioxide as a waste product. So carbon dioxide is a waste product of cellular respiration. Carbon dioxide outside of the cell is low because we're producing high amounts of carbon dioxide, so we have a concentration gradient leaving out. Therefore, our carbon dioxide dissipates out. Okay, now, is there a problem with this? There's oxygen there and carbon. Is this polar or non-polar? Why?
It's a linear molecule and it's being pulled the
Yeah,
same way.
the two oxygen. This is almost in a straight line, and those two oxygens are pulling equally, and therefore they cancel out. So it can get across the membrane quite well. All right. Another transport mechanism, just simple diffusion will not work for things that are large polar molecules. okay? They can't get across. But it's very obvious that cells do transport those things across. So there's another mechanism called facilitated diffusion. It is still requires concentration gradient. So the solute has to have a high concentration on one side, low concentration on the other. This is still passive in that the cell does not expend its own energy sources to accomplish the movement. But it is facilitated, okay? It requires a helper, a transport protein, okay? So this wouldn't work with a cell or a membrane that was just phospholipid. It has to have those embedded proteins. And so some of these embedded proteins, they're all transmembrane integral proteins, are called transport proteins. That's what facilitates the movement. So the molecule, the solute, doesn't go directly through the phospholipid bilayer. It goes through a transport protein, okay, that can allow it to cross. There's two categories of transport proteins. Some of these are channels, okay? So we have a protein in the membrane with very frequently, 7 alpha helices. Some of these channels have even more than that, but they create a hydrophilic pore through the membrane, right down the middle of the protein, so that it's a tunnel, so that the molecule can go right straight through. Just give a cartoon here, okay, like this. All right. Now this is a cartoon. Of course, this is a protein, but we have here our higher concentration here. They can be gated. Okay. What I mean for a gate is that they have 2 native conformations. One conformation closes that tunnel. Another conformation opens that tunnel. Okay. And therefore, once that tunnel is open, then these are free to diffuse through the channel. It's a water-filled channel. Very often, this part here near the gate is very, very, very narrow, okay? So that channels are not promiscuous, they're pretty selective, okay? For example, potassium, sodium, they both have positive charge, but one is a little bit larger than the other, and so a sodium channel will only let sodium go through, potassium channel only potassium but not sodium, okay? So they can be selective. There are other channels, however, that will allow both sodium and potassium, okay? So we have a variety of these things, all right? So these are gated channels a lot of times. So they would open the gate due to a conformational change, all right? Second, this type of transport protein are called carrier proteins. They're also called permeases, okay? Carrier proteins. In this case, there's no aqueous channel. There is a binding site on one side, and when the solute binds to the protein, it leads to a conformational change by which the solute is carried through the protein and dumped on the other side, okay? So it's carried across, all right? Okay, let me go back. I'll come back to that in a moment. Let's go to this, yeah, okay. Oftentimes these proteins, once again, this is a cartoon, but usually the binding site on one side is sort of funnel shaped, okay? Remember that this is about 8-9 nanometers, all right? But when the solute to be carried across binds, it's already almost halfway across, all right? And when it binds, it's binding. This is going to be a theme you're going to see a lot with proteins. Things that bind to it often lead to conformational changes, all right? So when this binds, it leads to a conformational change where it creates a funnel on the opposite side. And furthermore, the binding site where the solute has attached has changed so much that the solute can't attach there anymore. And therefore, it's released into the other side of the membrane. Okay, but this still requires a concentration gradient. Still requires a concentration gradient. All right, let me go back to where I was before. Yeah. Okay, so going back to the There are a group of special channels for water. They're universal, although from one organism to the next, the amino acid sequence of these aquaporins are different, all right? But it's still a family of proteins, and these are especially for water, okay? Now, I said water can sometimes leak through, but this is the mechanism by which most water will cross membranes. It's important to have water moving in and out of the cell. So these are aquaporins, all right? Now, I said there's some cells in tissues in the kidneys that they're impermeable to water, absolutely. And there's some that can be permeable, and they can change permeability. Once again, plasma membranes are always being refigured, reconstituted, changed. And so the way in which these kidney cells are doing it is by inserting or removing aquaporins. You remove all the aquaporins, there's no way for the water to defrost, okay? All right, so aquaporins, and that involves osmosis, okay? So osmosis, this is something I've already mentioned osmosis before, but you really need to get a good grip on it because it's biologically real important when it comes to semipermeable membranes. Water can move through aquaporins, but it does so down concentration gradients. Okay, yes?
Do they just, denature the protein and get rid of it, or
What's that?
for the aquaporins, you said that to control the levels
Of aquaporins?
Yeah, do they just,
Denatured?
yeah,
No, they're completely removed
what do they do?
into the cell, into a endosome. So it's not just an individual, you can't just grab a protein, pull it out, but you can have the membrane fold in as a bubble, okay? And so there's mechanisms to take these aquaporins, bring them into one spot, okay? And then the membrane folds in, we now have what looks like a transport vessel with the aquaporins that have been removed from the membrane, okay? All right, so osmosis. This is also facilitated, okay, because we have an aquaporin and the water is still moving from high concentration to low concentration, but usually when we talk about osmosis, we're referring to the solute concentration, okay? So this is a movement of water from an area of low solute concentration to an area of high solute concentration, assuming the solute can't move. Assuming the solute can't move, okay? All right? So where the solute concentration is low, the water concentration is high. Where the solute concentration is high, the water concentration is low. And so although this looks, when it says from a area of low solute concentration to high solute concentration, it's a movement of water, that almost looks like a contradiction when it comes to diffusion. But we're talking about the diffusion of water, not the solutes, okay? All right? Okay, yeah, this is a good model of a channel protein. And they can be pretty complicated in their structure. And so you see through here, these alpha helices in the green, there's a lot of alpha helices there, and they can shuttle just a little bit with conformational change to open and close. You're not looking at it from above, all right? This is from the side, okay? All right, now, these, both for channels and for permeases, these are usually pretty specific. As a general rule, they are specific, okay? So only certain types, both of them have binding sites. Not so much channel. Channel just opens and allows things to go through, but the carrier proteins, they have a very specific binding site for that solute, okay? So this is sort of like lock and key interrelationships, shape, counter shape, okay? If you have some solute that doesn't have the right shape, and I'm not just talking about three-dimensional space-filled shape, but also electrical shape, okay? Because where the polar bonds happen to be, they have to have a good match, okay? Or else it doesn't open, nothing will go through. Okay, furthermore, and I'm mostly talking about carrier proteins, to a lesser extent for channels, can also be true, their action can be saturated, all right? So if we took a graph and we increased, all we have is a phospholipid bilayer, and we have something that can cross the membrane by simple diffusion. If we increase the strength of the gradient, okay, making it real high on one side, real low on the other, and measure the rate at which molecules, solutes are moving across. Of course, you increase the concentration gradient, you also increase the rate. Here, the rate might be number of solute molecules per square micrometer per second, okay, unit time. And, but with carrier proteins, if we increase the concentration gradient, yes, initially, it does increase, then it starts to slow down, and then it becomes level. And any further increase doesn't improve anything. There's a maximum rate here, okay? Why do you think that is? Why do you think it would asymptote out?
Because there's only so many proteins that need.
There's only so many proteins, okay? This is like checkers in Safeway, okay? If you have 10 checkers, you have 10 checkers, but only 2 customers per minute. Let's say the checker can check you through in 1 minute, okay? Unheard of, but still, let's just assume that. If we have 10 checkers and 2 people coming up per minute, anyone measuring the rate of customers coming out of the store outside would see 2 customers leaving per minute, okay? So we have 2 checkers working, 8 of them talking, okay? Now, if we increase the number of customers up to 8, well, now we have 8 of those checkers working, 2 of them are just talking, okay? And outside, you see 8 customers coming out. Make it 10. Now all of them are working. Make it 15. Make it 20. How many are going to be coming out the front door? 10. It's a maximum, okay? Now all of the checkers are working as fast as they can. All right, so that's what happens here. We do have a limited amount of those proteins. We can't just fill up the membrane to the brim with proteins. There's a limit as to how many of these carriers can be there. Furthermore, things can go through channels much faster than carrier proteins, because here we have to have binding, conformational change, dumping other side, switching back to the original conformation, and that takes, although we're talking about nanoseconds, it still takes time, all right? Much more than a channel that just opens and things can go through it, okay? All right, so this can happen. Furthermore, both of these, channels and carriers, can be inhibited by a mimic molecule that looks like, in such a way, to the binding side of the carrier. So it might be that we have a mimic that isn't our molecule that we're running across, but if it binds to the binding side, the carrier protein might or might not be able to carry it across, but even if it could carry it across, as long as we have a molecule there that is not wanted, our desired solute can't get through, okay, and therefore it slows it all down, all right? Some of these will bind somewhere else and just completely alter the whole protein's conformation so that nothing can get through, okay? This is a fact. This is something we'll come back to a little later, inhibitors, all right? Okay, third mechanism is active transport. So, active transport, the cell has to use its energy sources. Okay, for the other two, simple diffusion, facilitate diffusion, we have to have a concentration gradient, but the cell is not using its energy resources to move it. With active transport, it does require the cell to use its energy resources. And it can, and usually does, move solutes up a concentration gradient. Most of the time is against the concentration gradient. There are some cases, however, Usually the textbooks always say it is always against concentration gradient, and that's not so. Most of the time it is, but you can still have active transport when it's down a concentration gradient too, okay? It happens, yes.
Would that be for like the bigger molecules, or would that be?
No, we're talking about the same size molecules,
Same size.
okay? Because these molecules have to be small enough to be able to go through a protein, okay? So if it's a big thing, bigger than the protein, it's not gonna get across that way. All right, so this requires ATP, usually. There's many different types of energy sources in the cell, but in general, ATP is the major, and or a concentration gradient, okay? A concentration gradient, ion gradient that the cell had to expend energy to create, okay? So this is the major difference here between this active transport concentration gradient versus facilitated diffusion, is that the cell had to expend energy to create the gradient, all right? Okay, so this is only carrier proteins. No channels are involved. No channels are involved. Only carrier proteins are involved in active transport. Okay, and I'm going to cover two different broad mechanisms for active transport. One is ATP-driven pumps, and the other one is something more complicated called co-transport. It is also called secondary active transport, because the active transport is not direct, okay? And I'll talk about ATP-driven pumps first, because they're easier to understand. Okay, so with ATP-driven pumps, ATP is the energy source, okay? But we have, okay, I'll come back to this in a moment. So here we have the pump. Carrier protein has a binding site, but we have higher concentrated solute inside than outside. And so just simply binding, unlike facilitated transport, diffusion, just the binding of the solute doesn't initiate a conformational change because we have a gradient pressure pushing back, okay? So molecular gradient pressure pushing back, and therefore conformation does not occur. So what has to happen is the protein has to be phosphorylated, okay? Usually these transport proteins are also ATPases, okay? When I say ATPase, it means that they can use ATP in ATPase. They have a binding site for ATP, and they can hydrolyze that phosphate, terminal phosphate. And so what it does is it'll attach phosphate to a very specific amino acid residue, R chain. Usually these are things like tyrosine or serine. You don't need to know that, but they have a hydroxyl group, okay? Usually a hydroxyl group. So by attaching this, that alters conformation. It elevates the energy content of the protein. Because remember that phosphate bond contained chemical energy. And when it's transferred onto the protein, it now has a higher energy state. And what we have left over now is adenosine diphosphate, which just only has 2 phosphates. All right? So just by attaching that, it leads to protein toggling into a new conformation, which dumps our solute on the inside of the cell, okay? And also this binding site for the solute has also changed shape so that our solute does not bind there anymore. Now to get it to go back, because these things aren't just used once, they cycle, they go through again and again and again. And so to return it to its original condition, that phosphate has to be removed, okay? So there are enzymes that are always there, they're constituent, and they will cleave off the phosphate, and this will toggle back to its original condition. So this is the cycle that it goes through, all right? So these enzymes collectively, I'll put it over on this board, are called Now, for this phosphate to get back onto ATP, it requires input of energy, right? It can't just pop back on. And this is the whole role of cellular respiration, to regenerate ATP from ADP plus inorganic phosphate. We'll get back to that later. Okay, so an example of an important ATP-driven pump is calcium ATPase in smoothie R, in muscle, okay? I already mentioned that calcium is very important for muscle contraction. If there's calcium in the cytosol, then muscle fibers can contract, but in order for them to relax, we have to take the calcium back out. And so it has to be pumped into, against the concentration gradient, back into the smoothie R. So if we have here the smoothie R, then there is calcium ATPase, it does require ATP, but it will pump in one cycle, 2 calciums into the smoothie R, right? Okay, now, give you another example of an ATP-driven pump that you need to know very, very thoroughly. It will come back again and again and again, even in Biology 101B. In fact, you'll hear a lot about it in Biology 101B. And this is called the sodium-potassium ATPase. This here calls it sodium-potassium pump. That's what your book will call it. But it is a more complicated type of ATPase that carries both sodium and potassium. And so with one cycle. So with one cycle, on the inside, there are binding sites for 3 sodium ions. So these will bind on the cytosol side. Okay. These are usually in the plasma membrane. Okay, they're located in the plasma membrane. So 3 sodium will bind. The concentration across all cells, sodium is high outside, potassium is high inside. They're not in equilibrium. Okay, so potassium is high inside, sodium high outside. And so we're wanting to move the sodium to the outside against the concentration gradient. Okay, so that's just not going to happen unless we phosphorylate. Okay, so by attaching a phosphate, that leads to the conformation will change. And so this toggles into a new conformation by which the sodium are carried across the membrane, through the protein, through the carrier, and dumped out here. Because now those binding sites for sodium are no longer in the right shape to hold sodium. But what shows up are 2 others that are binding sites for potassium. So potassium now can bind on that side, 2. So 2 will bind, and then we remove the phosphate. This causes it to toggle back into the original condition, okay? Which would dump our 2 potassium on the inside, okay? So one whole cycle of sodium potassium ATPase carries 3 sodium out, 2 potassium in, okay? And all cells have a higher sodium concentration outside and than what they have inside, and it's just the opposite for potassium, okay? This is very important. The energy requirements of kidney and brain, they have a lot higher energy requirements of ATP than a lot of other organs in your body, and most of that energy is used for sodium potassium ATPases, okay? All right, okay, so that is a very important pump. Co-transport. This is also called secondary transport, secondary active transport, because it sort of combines active transport and facilitated diffusion. However, it is not either of those. It is a mechanism all of its own. And so what it does it here, one thing, we do have to have an ATP powered pump that pumps ions across the membrane, creating a ion gradient, a concentration gradient, okay? Could be a sodium, could be hydrogen ion, etc. But so it's using its energy to create a gradient. And then there is a totally different protein, carrier protein, that allows that first solute, which is the energy source now. This is the energy source, all right? It will pass through this carrier protein. It has a binding site not only for solute 1, but also for another solute that we are moving against its gradient, the second solute. They both have to bind. And so it is the energy of the first solute that leads to conformational change that drags the second solute across the membrane through the carrier protein into the cell against its gradient, okay? It's called cotransport. I'll give you an example. In plants, okay, they have a tissue through which sugars happen to be transported, and sucrose that's being produced, first of all, was glucose that's produced in photosynthesis, then it's arranged into sucrose, and then that is what is transferred through the plant. So sucrose has to be loaded into the cytosol of those transport cells. Our concentration of sucrose is high here compared to what is out there, and therefore we can't use facilitated diffusion. It has to be carried in against the concentration gradient, but it isn't direct. What we also have there is a hydrogen ion ATP driven pump, okay? So it has to use ATP, but what it's doing with the energy, and this is once again a carrier protein, it is moving hydrogen ions to the outside of the cytoplasmic membrane. So this is creating a high concentration of hydrogen ions out here. And so we have a concentration gradient, energy source. But there's something else here. We have two forces. We're also moving positive charge out. So if we have excess more positive charge outside than inside, we have electrical gradient here too. So along this membrane, the outside is positively charged and the inside is negatively charged. Positive charged particles will move toward negatively charged areas, okay? So we have two forces here, not just only a concentration gradient, but also a charge gradient. That's referred to as electrochemical gradient, okay? So we have a carrier protein. This is the cotransporter. So this consists of a ATP-driven pump and a cotransporter. So with the cotransporter, both sucrose, and there's a binding site for both sucrose and a binding site for the hydrogen ion, but it is the electrochemical gradient of the hydrogen ion wanting to get back in that leads to the conformational change and drags the sucrose against its concentration gradient. Okay? All right. So this is cotransport. The reason it's called secondary active transport is because the actual transport is one step removed from the ATP-driven pump. Now I mentioned how the wheel protein, okay, wheel protein in driving the flagellum in bacteria, prokaryotes, okay, they have a hydrogen ion ATP-driven pump also in their membrane, and they're pumping hydrogen. They have to use ATP to move hydrogen ions out, and that is then the force that drives the wheel protein as the hydrogen ions come back in, okay? All right, so this is electrogenic, okay? It means that the pump is producing a charge gradient. The ATP, or the sodium potassium ATPase, is also electrogenic. It's creating not just only a concentration gradient, but also a charge gradient. Why? It's moving a positive charge out and positive charge back in. Sodium out, potassium in. But it's moving 3 positive charges charges out and allowing only 2 to come back in. And therefore, there's accumulates more positive charge outside than inside, and so it's electrogenic. Any other pumps that are maybe, or even carrier proteins, if they are exchanging, because there's some that will do this, we'll have a concentration gradient of a positive ion here, and we need to bring another positive ion that's on the outside in. And so this is moving out, 1 positive, and it's bringing in 1 positive. We're creating concentration gradients, but it's non-electrogenic. Okay, there's no charge difference coming about. All right? So these electrochemical gradients, as I said, they have 2 factors, 2 things. A chemical effect. Chemical effect is concentration gradient, period. Okay? They also have an electrical effect, that is a charge difference. All right? And these can be oriented in different ways. All right? Now, if it happens that we have the charge gradient, electrical gradient, and the chemical gradient, the concentration gradient, and the charge gradient going in the same direction, then transport can be pretty fast. Okay? So if we have here a pump or a co-transporter, or even a carrier protein for that matter, for sodium, okay? Have a high sodium concentration here, sodium ion. We have low concentration here, so that favors crossing the membrane. But also, if this side of the membrane is negative, this side is positive, moving a positive charge into that negative area is also favorable. So this electrochemical gradient will work just fine. But these 2 can be opposed. They can be going in opposite directions. Here, then, the direction of transport will depend on which one of these 2 forces is greater. Okay, so here I'm showing one where the concentration gradient is greater than the charge gradient, and therefore we still get transport because the chemical gradient is stronger. So we still get the net change across from left to right. However, it will be slower than this type of scenario. Or we can do it just the other way around, where the electrical charge gradient is stronger than the chemical gradient, and once again, we will get net movement, this time from right to left, but it will be slower than the other phase. Okay, so there's 2 components to these electrochemical gradients. All right. Now I'm going to give you a real life example here, which is going to combine all the stuff I've said about transport. Okay, all together. Okay, when you eat a meal, it's macromolecules. It goes into your stomach, then into your large intestine, and all those macromolecules have to be broken down into monomers, to basic building blocks. They're transported across one layer of cells that lines the lining of the small intestine. This particular tissue is called an epithelium. One layer of cells, and these are sort of column-shaped cells. What is significant about epithelia is that these are close-packed cells. Forming a sheet. The plasma membranes are touching each other. Okay, furthermore, there are proteins, if we're talking about vertebrates, that go from one plasma membrane to the next, all the way around. These are called tight junctions. And what the whole point here is that if in other tissues, we can have cells that are relatively close together. And if we have concentration gradients, solids can still move between. There's no barrier. But with these tight junctions, it creates a seal so that nothing can go in between the cells. Significance of that is that the cell has control over what crosses that tissue. Okay? All right? So this is what I'm showing you. This is the gut, this is the lumen of the small intestine, okay? And this is an epithelium cell, okay? This is epithelial tissues. We have tight junctions here that hold them together. Furthermore, epithelial tissues have some other quality here that's unusual for cells. This is called the apical side. And way over here is the basal side. And they're polarized. They're not the same. They don't have the same proteins in them, okay? So other cells, typical cells, the plasma membrane is the same all the way around. You have the same types of proteins everywhere, okay, for the most part. However, this here has what's called apical basal polarity. Okay, the apical side is different than the basal side. Now, this is one layer thick, and there's something here, fluid-filled area, and then right here is a capillary bed, capillary, okay? So to move something into the bloodstream, it has to cross the apical membrane of the cell, be brought through the cell, cross the basal membrane into what is called interstitial fluid. Interstitial fluid, and it goes into a capillary. Capillary is one cell layer thick. One cell layer thick, all right? And then it goes away, all right? So this is how stuff gets into, from your gut into your bloodstream. It has to cross 2 layers of cells. 2 layers of cells. I'm only concentrating here for what happens with the epithelium, okay? All right, so you eat something and we have glucose, okay? Now, it takes a while for starch to be completely broken down to glucose, but you still have some free glucose in the food that you eat, and we don't want to waste it, okay? So if we have here just a little bit of glucose, and the concentration of glucose inside the cell is higher than outside, we can't have facilitated diffusion, right? Okay, because it's against the gradient. So what happens here? All right, let me point out some players here. Our sodium potassium ATPase, which I've already talked about, it is only in the basal membrane. It's not in the apical membrane. And in the apical membrane, we have a cotransporter. A sodium, it has a special name, but for this class, we're just going to call it sodium glucose cotransporter. Now, let's just make an assumption here. First of all, our sodium concentration, we have 3 compartments that are separated from each other. We have the gut lumen, we have the cytosol of the cell, we have the interstitial fluid. 3 different areas that are separated by membrane, okay? Let's say that the sodium concentration here, here, here are initially the same. Initially the same, okay? We're going to have our sodium potassium ATPase. Wrong button there. So here we have glucose high, glucose low, but we're going to let this critter spin. We're using ATP. We're pumping sodium out, potassium in. We're going to ignore the potassium, okay? We're just looking at the sodium. What's going to happen to the sodium concentration inside the cell? First of all, in all 3 compartments, we start with sodium being the same. We're pumping sodium out of the cell. What's going to happen to the sodium concentration inside the cell? It's going to go down. We now have a concentration gradient across that basal membrane. Low inside, high outside. But what about across the apical membrane? Is there a concentration gradient here? Yes, because if we pump sodium out here, that's going to be low. This is going to be high, right? Therefore, we have a favorable gradient for sodium, but it's not favorable for glucose, okay? So now,
So we have sodium high there, sodium low, sodium high, and so we can, this is a co-transport. We've created a concentration gradient using ATP from sodium, for the sodium potassium ATPase, and it can, so our energy source here is the ion gradient. Sodium ion gradients carrying this across and bringing glucose in, okay? So now we have high glucose inside. And of course, the sodium doesn't accumulate because we just keep pumping it out. We keep pumping it out. But now that we have high glucose here, low glucose over here, we can use facilitated transport at this point. And so there is a GLUT2 for facilitated. This is a protein, carrier protein specific for glucose. We have several different varieties of glucose transporters in different parts of the body, and they're related proteins due to gene duplication. But this here is GLUT2, and therefore our glucose just goes across here. All right? Now, this will go into the blood. Our glucose will not accumulate. Our sodium concentration here will not accumulate because both of them in the interstitial fluid is higher concentration than what's in the capillary. They go into the capillary. And the blood is moving away. And so that stuff keeps getting carried away so that we never accumulate a high sodium concentration or a high glucose concentration on that side, right? So this is putting together a lot of stuff, transport. Now, let me ask you a question. If membranes are fluid, how come this can only be here and these two can only be here? What keeps them there? Membranes are fluid. Things move laterally. How do we keep them there? Partly the tight junctions prevent the fusion around, but there's also something else that can happen too. That's one thing. But it is tight junctions, yeah. What else?
Cytoskeleton?
What?
The cytoskeleton?
They could be anchored to the cytoskeleton. Okay, so both of those mechanisms happens. Okay, now to put more complexity here together, I mentioned always that membranes are constantly being changed due to conditions. As we digest our starch, okay, starch has 10,000 glucose units. We eventually get this high concentration. Okay, glucose high, higher than inside. And therefore, things can change, all right? So now we're gonna have here, now we're still keeping our sodium potassium ATPase, it's still working, but now we have, after digesting the starch, we now in the gut lumen have high glucose, low compared to what's inside, okay? So now that's favorable for facilitated transport, but we don't have any facilitated transport there. Now this difference in glucose concentration is sensed by the sodium glucose co-transport. And we don't know exactly how the signal gets through, but nonetheless, what will happen is that GLUT2 will be inserted into the apical membrane. Okay? That difference in glucose concentration is sensed here. There is a signal transduction pathway, we'll talk about that later, that communicates with a transport vesicle, which was anchored right under the plasma membrane. Okay, so if I go back to, because I can't write on that, if this down here is the lumen, no, this is our cell. This is the lumen side. Okay, this is the apical membrane. We had tethered to cytoskeleton in this area, transport vesicles that have GLUT2 in them. Okay? GLUT2 was made on a rough ER, inserted into the membrane of the rough ER, transported to the Golgi through a transport vesicle. It went through the Golgi, came out as a transport vesicle, but instead of fusing to the plasma membrane, these are just hanging out there until they get the right signal. Then they fuse. When they fuse, they insert GLUT2 there. Now we have a favorable gradient. And now we can just use facilitated diffusion to bring the glucose in. And since we have high glucose there, it just goes right out into the blood. Okay? And then what happens when our glucose levels drop again? Well, that part of the membrane that has GLUT2 in the apical membrane, they're brought back in by invagination. Okay? This is the way life is, isn't it? Fantastic. Really, when you think about it, this is really, really, life is fabulous. Okay? Think about it. Okay, any questions? Okay, all right, so these co-transporters, two different types. Okay, let me just summarize that. With co-transport, there's two things that happen. There's an ATP-driven pump that is the cell uses its energy, ATP, to create electrochemical gradient. Okay, and then the electrochemical gradient is now an energy source, and so it can that gradient can be used to carry another solute against its concentration gradient. Okay, now I've been showing here two different solutes, but some of these co-transporters get more complicated. There can be one that is an energy source, and it drives the transport of 2 or 3 other solutes. All right. Now these can be divided. These co-transporters. So this is referring to co-transporters. Symport is a co-transporter that's carrying, but that all the ions involved are going in the same direction. But there are also antiporters. And the antiporter, they are moving in opposite direction. Okay, like this. All right. Now, I want to emphasize here, at least in this class, this word symporter and antiporter are often used for any sort of carrier proteins, even for ATP-driven pumps. However, in strict physiology, the big boys, you get a good big fancy physiology book, cell physiology, symporter, antiporter are only for co-transporters. So our ATP, our potassium, sodium potassium ATPase is not an antiporter. It is transporting sodium and potassium in opposite directions, but it's not a co-transporter. It is an ATP-driven pump, right? So these terms should go only for co-transporters. All right, now there's still things that get bigger, all right, such as big proteins. If you have big proteins that have to be brought in from outside, for example, macrophages, these are parts of your immune system, they eat up whole bacteria. Okay, if you get sick with a bacterial infection, they will bring that whole bacterium into the cell and fuse a lysosome to it and digest it, all right? That's too big to cross through a protein because it already is made of hundreds or thousands of proteins. And so to get that in, it involves infolding, outfolding of the plasma membrane, okay? There's something called bulk flow, endocytosis, exocytosis. Now this particular figure has different types of endocytosis. I just want you to know endocytosis, period, okay? So this is involving movement of material from outside of the cell. It collects along the surface of the plasma membrane, and the plasma membrane folds inward and buds off a bubble, which is, it looks like a transport vesicle, but I've been calling them endosomes, okay? And so most of the time, those things end up fusing with a primary lysosome, okay? So that's, up here, it'll have different types, and I don't care that you know about receptor-mediated endocytosis and pinocytosis, phagocytosis, etc. Just endocytosis, moving stuff in. Okay, involves the whole membrane. Just the opposite, taking thing out is exocytosis, and we've already referred to that. How do antibodies get out of the cell? Exocytosis, transport vesicle fuses to the plasma membrane, dumping things out. Okay. this also involves energy, but we usually, when we're talking about active transport, we're usually restricting that to ATP driven pumps and co-transport. Yes, this does involve energy, ATP and other things. It also involves all sorts of proteins that will fold the plasma membrane, okay, that are involved there as well. It's a complicated process. So a lot of the cytoskeleton and other types of proteins that will bend membranes and allow them to fuse happen to be involved. Okay, those are transport. Now I'm going to switch to another story, okay? Any questions first? Any questions? Okay, all right. Membranes, especially plasma membrane, is also involved in communication, not just transport. Okay, now I'm switching from transport mechanisms to a new story, communication. Okay, so cells communicate with each other by releasing chemical signals, and only those, there's various types of chemical signals. Some of them are, well, in multicellular organisms, cells have to communicate with cells, but even unicellular bacteria still communicate with neighbors around them, okay, so they coordinate activity. And so they have to be able to read those signals. So usually this is a solute, and it has to bind to a receptor on the surface of membrane. And this leads to what is called a transmembrane signal that goes into the cell, and it changes the behavior of that cell. Some of the things that can happen is that some of these signals can just say, you're fine, don't change, just stay how you are, you just don't move. Other times it might be signals that tell, hey, you're getting too fat, it's time to divide up, all right? So it'll divide, all right, and grow. Signals might say, yeah, you're not doing too much in the shape you're in, I want you to do something special, okay? So it changes its cell type completely, okay? Differentiation, all right? Or, and sometimes these signals might say, just kill yourself, we don't need you anymore, all right? We do have those, all right? So they do have them. And so these signals is what I want to talk about. How do they work? So some of them, the cell that is signaling, it is releasing, those chemicals just don't pop out of anywhere. So there's special types of molecules that are released by a signaling cell. If this is hormones, we're talking about a gland, okay, that will release this hormone into the blood. But even cells that are fairly close together may communicate by cytokines, okay, something that they release. That's mostly in your immune system. That's how your immune system will communicate among immune cells. We'll get to that at a later time, or hormones, okay? Now, the target cells, that is the ones that can respond to it, they have to have the receptor. If they don't have the receptor, they cannot perceive the signal, okay? And so a lot of hormones, for example, there's a hormone that's released from your pituitary gland. Pituitary gland is right under your skull and under the brain, gets into the blood, but the target are certain cells in your kidney. So that is put into the bloodstream, and all the cells in your body are exposed to that hormone, but only cells in the kidney have the receptor, and they're the only ones that will respond. So if you don't have the receptor, you don't see it, okay? So, and that is almost always protein. So this is a cartoon that's sort of showing that it's happening. All of these receptors are transmembrane, okay? So they have a binding site, and this binding site is very specific shape, counter shape interaction, okay? If you have a hormone or a binding site that differs from the hormone that's circulating, they won't do anything, okay? It has to be a match. So when this binds, it leads to conformational change all across the protein that is communicated to the other side. And this might be a catalytic unit, might be enzymatic, and it will cell's changing the whole program that the cell is doing. Okay, these are called signal transduction pathways. Signal transduction pathways. I'll have it up later. Okay? Ligand, you'll see that word once in a while. A ligand is any sort of molecule that binds to a protein and leads to action on the protein's part. Here I'm talking, so this is a broad word, okay? But specific ligands might be hormones. Specific ligand might be cytokines and so forth. Okay, so that's just a general. All right, so this will lead to the behavior and response of the cell. Now, I'm going to go through some types, categories, of receptors. One type are called ion channel linked receptors. I'm not going to go into too much talk about these right now, but when we get to Bio 101B and talk about how neurons work, then we're going to come across these a lot. Yes?
Can you just go back to the slide?
Okay. Something you can touch on.
Yeah, it's.
I was wondering, something's wiggling here. What's going on here? Maybe.
Wow.
That was annoying. I thought, I need to walk lighter, because it sounded like the screen was wiggling. Right? That's what I heard. What did it sound like to you? Yeah, yeah. See, I can't hear, high sounds. And, I know one time, the students were just cringing, and what's wrong with you? And they said, can you stop that buzz? I didn't hear a thing. And I called some other old guy in who, works with the media, and he was, I don't hear anything, okay, but we shut it off and people were happy. All right, but I heard this one, okay, it was starting to get annoying, all right? Okay, get back to this, behavior of the cell. Okay, so ion-linked receptors. These are really plentiful, for example, in the nervous system. So the receptor controls an ion channel. A channel protein, okay, gated. And so the binding of a signal simply opens the gate. Okay, that's all right? And we'll come across those a little bit later. Two others that are a little more complicated, enzyme-linked receptors. Okay, usually with this signal transduction, any sort of signal transduction, there is a receptor and an effector. The effector is what responds to the conformational change in the receptor. There's going to be a third item here in just a moment. So in the case of enzyme-linked receptors, the receptor is both. It is both the receptor and the effector, okay, at the same time. And so usually this is an enzyme, okay, or the receptor is very closely related to an enzyme. But most of the time, our receptor is an enzyme as well, has a dual functionality. And usually that effector is a kinase. Now, protein kinases, this is a broad group of family of enzymes. And what they catalyze are phosphorylation reactions. So what does that mean, phosphorylation? Phosphorylation, what is phosphorylation? It's attaching a phosphate to a very specific R group on a protein, which will lead to a conformational change in that protein, which will change what it does, turning it on or turning it off, all right? So we're talking about thousands of different types of protein kinases, okay? Some of them, they're all very specific for a particular type of phosphorylation reaction. therefore they're also using ATP as the phosphate source, all right? So they're important. So now this will lead to either activation or deactivation of proteins inside the cytosol, all right? And and the cells are what they are doing what they're doing due to the interactions of those proteins. If you change their activity, it also changes what's going on, okay? All right, so I'm going to give an example of a enzyme-linked receptor. This is called tyrosine kinase. This, once again, is a group of receptors. It's pretty common, but we have many, many different subtypes. Tyrosine kinase, it is both the effector and the receptor. Usually these are monomers in the plasma membrane, meaning that they're not attached to each other, okay? But when they are active, they dimerize. They have to fuse together. And so what we have here is a binding site, and when a signaling molecule will bind to two of these, it leads to a conformational change, which makes them sticky and attracted to each other. They would dimerize, okay? So there's two of these proteins together, and the conformational change, they put the ATP up here, but it really happens down here. Why this is called tyrosine kinase is because there's an amino acid, 6 different tyrosine residues. Tyrosine is an amino acid, so it's part of the structure. And these become phosphorylated using ATP, okay? That doesn't happen up here, it really happens down here. And that leads to further conformational changes of this effector site. And that will attract other types of proteins, and it leads to a cascade of events that changes the behavior of things inside the cell, okay? So this is all I'm going to say about tyrosine kinase. It is the most popular. There's other enzyme-linked receptors, but this one here is pretty widespread, okay? What's more widespread even than that, and we will see when we get to physiology and use of this quite a bit, is G protein-linked receptors. A lot of them. Instead of having just only a receptor and effector, which are separate from each other, okay? With tyrosine kinase, the receptor, effector were the same, okay? Here they're different, but there's also a relay protein between them, all right? So this is, in general, cartoon from your book. This is the G protein receptor, but instead of using this, I'll come back to it. I want to use, come back, all right? I'll come back to this in a moment. All right, I didn't know I had all that. I want to use mine. I like my drawings better, all right, okay? So if we have here a receptor, there's a stimulatory hormone. It binds, it leads to conformational change of the receptor. Now the relay is something that's called a G protein, okay? It is a trimer, 3 subunits, 3 proteins hooked together, okay? And why it's called G protein, this is a guanosine binding protein. Guanosine is a nitrogen base, okay? Now, not only do we have ATP, there's also GTP, okay? Guanosine triphosphate. And so it's just like ATP, but all that's different is instead of adenine, it has guanine, all right? And sometimes this can act like ATP, being an energy source, but that's not what it's doing here. It's just a trigger. Now, when this G protein has a binding site, one of these subunits has a binding site that will house GDP, guanosine diphosphate, okay? And when that is bound, guanosine diphosphate, the G protein is inactive. The G protein is inactive. So once we get, and these are usually close to the receptor. So once we bind the signal, we have a conformational change, and that will affect the G protein, that its conformation changes too. So that GDP is kicked off. The binding site changes, so that GDP cannot bind, and it's changed into a binding site for GTP. And GDP binds, okay? No phosphorylation happened here, it's just an exchange, okay? And once GTP binds, then one of those subunits, the one that has a binding site, will detach and move along the membrane until it binds to the effector. And there's different effectors, but this effector is adenylate cyclase. There's others. That turns this on. It was inactive, okay? Because once again, it binds something, leads to conformational change, and what this does is it converts ATP to another form called cyclic AMP. Okay? All right, it's a little bit more complicated. Let me go show you the difference here between these different molecules before I come back here. Okay, up here is what ATP looks like. If we adenylate cyclase, it's also adenyl cyclase, same thing, cleaves off two of those phosphates of ATP and creates another bond. This is what makes it cyclic, and this is cyclic AMP. If this bond is broken, okay, at the number 3 carbon, then we have just simply adenosine monophosphate. A little difference, very little difference, but it really, that little difference makes all the difference. This here is a signal, this is not. This is a signal, that is not, okay? And so what we have here is that cyclic AMP is what's called a second messenger. What is the first messenger? It's our hormone on the outside, okay? That's messenger number 1, and it leads to the creation of a second messenger that binds to a group of proteins that are called cyclic AMP dependent protein kinases. They're usually always a kinase, okay? So what's happening here? This is an inactive protein, but it has a binding site for cyclic AMP. When cyclic AMP binds to it, what happens? Conformational change, turns it on. And it's a kinase. What do kinases do? They phosphorylate. So it phosphorylates very specific other proteins, either turning them off, turning them on. And it changes everything that happens in the cell. There's more to the story, but I don't have time to do it. So I'll leave you in suspense. How do you shut it off? Okay, because you have to shut it off. It can't go on forever like this, okay? If it keeps going, it's permanent, okay? I'll be worried about it over the weekend, right? Okay, so, quiz for Monday. Where did I start Tuesday? I don't remember. I know where I stopped today. Okay, it's going to be here. But where did I start?
The MS
What?
endos chloroplasts and mitochondria.
Ah, yeah, chloroplast and mitochondria. That's right. Okay, so after the chloroplast and mitochondria,